Next speaker is Stephanie Wankowicz, who is a assistant professor in the Department of Molecular Physiology and Biophysics at Vanderbilt University. So,
Awesome. Yeah, this is like stand. Sorry. Thank you so much for the invitation. I really enjoyed this session last year, and I was really happy to come back and talk. And so, you know, I am going to talk a bit about sort of like the ensemble modeling, the sampling problem, and sort of the experimental metrics that we face. I don't have, you know, too many, I think, solutions totally in this space, but I think it's clear themes that a lot of us are thinking about, and sort of like, how do we approach this so that we can use these different types of foundation models to actually be able to build up, and what we're really interested in, how to actually fit experimental data better, right? So proteins are incredibly dynamic molecules. They're constantly wiggling and jiggling. And the majority of structural biology that we have reduces a lot of these dynamics to a single structure. So the models that we get are single structures. And what these single structures have enabled us to do is have really great benchmarks for us to do structural predictions. And, you know, there's been decades of work on these metrics, on how do you actually develop metrics to be able to get these, right? AlphaFold came out, obviously, because it has a ton of data in the PDV, but it also came out because there were years of how do you actually develop metrics that can appropriately compare between static structures and predicted structures. And with ensembles, we don't quite have that robust metrics, and we don't necessarily have these robust models, although the data contains a lot of ensemble information. Right, so if we want to predict ensemble models, right, what exactly are we trying to predict? You have information from lots of different experimental areas. You have lots of information on, you know, is it appropriate that you have to get every single Boltzmann weight exact? Do you have to just get sort of approximation? Is it the fact that we want to discover drugs? Is it the fact that we want to understand how enzymes work? Is it that we want to understand how cellular models work, and where sort of are these? And then when will we know how we've succeeded if we don't necessarily have this very rich sort of data and benchmarks? And so if we sort of think about some of the things that are preventing us from getting there, and I think a lot of the ways that the structural biology community really needs to sort of step in and help fill in some of these things, even if you aren't necessarily building the ML models, right, is that one, structural ensembles are really inconsistently defined across disciplines. So we've seen, you know, from SACS data to sort of what we're interested in, right? You can get different conformational states that have different time and length scales. So looking at these large domain motions down to very small atomistic movements. But at the end of the day, to sort of translate this all to biology, right, they're all interconnected. So they all depend on each other, they're hierarchical, and each sort of piece relates to different functions that the proteins are doing. There's also a piece that, right, no single experimental technique alone can fully capture these ensembles with the accuracy and the precision that we need to reflect their behavior in vivo. And so we, you know, jumping off of an incredibly rich history of sort of the integrative structural biology community, thinking about how do you combine information from different experimental data, how are we thinking about this from, you know, very just, you know, the raw experimental data, and also thinking about this in terms of statistical structural biology. So how do we actually leverage the fact that we have lots of structures of the same protein and information, and can we leverage that to be able to extract out conformational ensemble metrics? And then we have, three, which I think is a lot of the focus currently, and is very much the focus of our lab, is thinking about the fact that experimental approaches for determining ensembles still face some really major challenges. So all of the data that you're getting is ensemble averaged. You have data sparsity, so the states that you want to actually look at, oftentimes these excited states are in the data very infrequently, and then there's intrinsic measurement errors that you're getting at. And so if we think about sort of, a toy sort of input data, you're going to have some protein that has 8 different states. And what happens is all of the data is ensembled, and then you can get out a model. And so even if this model perfectly explained the average data, it's not necessarily going to match the input. And so how do you actually fix that? And so going into sort of these ensemble average data, it also emphasizes really why we have to model these as ensembles. So when we collect X-ray and cryo-EM data, we're not getting information on a single protein or a couple proteins. We're getting it on millions of proteins. And so how do we accurately model all these states, importantly, without overfitting the data? And then really thinking about how we deal with 2 types of heterogeneity. So the first, what I think we've been talking a lot about today, is conformational heterogeneity. So we have it when 2 components occupy multiple positions. And then you have compositional heterogeneity. So in X-ray, a lot of times this is a small molecule that is only in certain copies of the protein. In EM, it can be much larger components. But what gets really tricky, right, is that oftentimes these are almost always entangled. And so if we think about sort of models that are not considering all of this information, you can lead to a lot of sort of that overfitting information. And then it becomes unknown how to think about sort of atomistic foundation models and interacting with this. And I think as Doris brought up at the beginning, right, this is all of the types of data that is going to be collected with this new open bind data set. So right at the Diamond Light Source, they're really great at and have really pushed forward a ton of this fragment screening data. But when you look underneath what the data that they're actually collecting, right, is going to be an enormous amount of compositional heterogeneity that usually leads to compositional heterogeneity. And so if we want to actually translate what they're collecting to being able to understand how protein ligand interactions are occurring and what this means in terms of affinity, we need ways to be able to disentangle this. And so we've developed QFIT, or helped develop QFIT. It's an optimization algorithm that identifies these multiple states that exist in cryo-EM and X-ray crystallography. So right in this data, oftentimes you can get relatively drastic changes of side chains and backbones that oftentimes are not modeled, mainly because this is done manually, and there are no automated ways to think about sort of this overfitting. So just very briefly, sort of how QFIT works. So it's all done in real space. We use mixed integer quadratic programming to determine the best fit between a map. So your real space information. And then we really want to try to minimize this. So we can calculate the estimates of the density from each conformation that we can sample. We'll get into sort of the sampling problems a bit later, and then we can weigh them by occupancies. And so for the most part, because of the ways that we are sampling currently, we weigh over sample. And so we rely on this optimization algorithm to really be able to zero out many of these conformations. And so this has worked, but it is becoming very computationally intractable. And so we also put different constraints on this problem to just make sure that we are not, that we're fitting to sort of what we believe is sort of real density. And then we also use a Bayesian information criteria to be able to fit in multiple of these positions of side chains into the electron density. So just sort of examples of what we're able to find. And so, a large part of what my lab does is say, hey, how can we go back into the PDB and be able to fit a lot of this information better, right? We can find these sort of hidden alternative conformations that in a deposited model oftentimes are not modeled. And so for those of you who have modeled X-ray or EM data, there are some of these conformations where you're like, okay, I probably could have found these in Coot and modeled them myself. But there's a lot more subtle, such as this sort of phenylalanine, that becomes a lot harder to be able to model individually. And so having algorithms to help sort of guide and change this really helps. And so, as I said, we've been, going through the PDB, and we can see that if we just use sort of this R-free metric, that QFit improves the fit to experimental data in about 80% of structures. And then we can also look at this in sort of like an RSCC basis, so looking at real space correlation coefficient. So anything above this red line means that we're doing better with QFit. And as you can see that there is, a quite more drastic bump up when we look at sort of this real space correlation coefficient compared to R-free, probably indicating that, a big thing that I know Minhuan and I are very interested in, which is sort of like the solvent problem with all of this, and how to actually include that in a lot of these models. We can also see that we improve model geometry. So looking at mole probity, we can see this sort of left shift of QFit models indicating that we fit better. And so if we think about this in terms of, fitting a single conformation of a side chain into data that requires multiple conformations, thinking about sort of squeezing a round peg into a square hole, you're going to strain the information. And so if we fit multiple conformations, not only are we improving the fit to the experimental data, but we are coming much closer to what we know is sort of physically the bond lengths of this physical information. And so, we can also sort of proof of principle have at least shown that we can do this in cryo-EM data. Okay, so, one of the things, as I said, that we are, very much coming to is the fact that the ways that we have been sampling is using very exhaustive sampling of side chains and backbones, but that's not really realistic in terms of computational complexity. And now we have a lot of other information. So we've been trying to go at sort of like, what are other models that exist? How does this actually fit to experimental data? So the first one that we took was just taking ensemble refinement, which came out of the Phoenix team 13 years ago at this point, and is still, trucking along really well in terms of modeling ensemble. And so just looking at the R-free between the ensemble and the redo, we're sort of at about almost like a 50-50 of whether you're improving R-free. And so we can take a lot of these and we can compare this to R-free of QFit and ensembles. And so what you can see is that we always, or in the majority of cases, we do a better fit of R-free compared to ensemble refinement. And this is like very much as we would expect, occurring at much lower R-frees. And this is really indicative to the fact that we're doing a lot of this in real space. And so if you don't have sort of a lot of that phase information at the beginning, we tend to just do much poorer fitting in QFit. So this is, not necessarily going, maybe there are pieces of this that we can tease out and use this information, but maybe neither method sort of that uses the experimental data is going to work. So then we turn to sort of other predictive methods. So we've been developing this ensemble benchmark pipeline. What we really took after this was thinking about You know, in MD simulations, we have been benchmarking against really high quality room temperature crystal structures and NMR structures to be able to iteratively improve these models. And so if these ensemble predictive models are doing similar things, we should sort of have this same continuous feedback mechanism that we can do. And so what we did is we took 5 existing both ensemble prediction, and then we also benchmarked against sort of 2 you know, more static, well, open fold is definitely static prediction, and bolts you can kind of decide if you're using MD or X-ray, and then say exactly how is this actually fitting to a lot of the experimental data, looking at real space. We use SF calculator to look at R-free, and then you can also look at sort of different metrics. And so using sort of an initial data set that we did to play around with, we just first were looking at sort of RMSF. And so what we can initially see is just in SAM2 and BioEmu, which are both, and well, AlphaFlow is also trained on MD data, that you can see this really uptick in RMSF. So your deposited are in gray. Most of these are multi-conformer models, and you can see that there is like quite an uptick. But this doesn't really give us all of this information on, you know, is it just sort of having much higher RMSF everywhere? Are we actually recapitulating where sort of the experimental data is telling us? And so we can go through and we can plot right along each residue line. How does the RMSF really change? How is it moving together? And look at this sort of cosine similarity of the RMSF across the data set, or we have some structures that have a much higher and better job at comparing between the RMSF of individual residues compared to others. But this is just sort of like the start of how we very much see being able to include a lot of these generative ML models that, you know, maybe you can improve a model, but you have to go back to this experimental data, use a lot of these models to fit the experimental data, and continuously think through here. Right, and so as I said, you know, a lot of the things that we're sort of thinking about now is, right, are these good enough, these pre-training models for sampling in the modeling that we want to do, or are they sort of fitting other things? A lot of them are trained on MD simulations, which are fantastic for some stuff, but are very limited in time scale. Right, where do they fail, and are there ways that we can sort of use the best of each method to be able to do this? And then what are the best metrics to fit to this experimental data? We have sort of real space correlation coefficients that can give us information on how it's actually fitting in local space. But eventually, I think for those of us who are interested in, you know, how do we get a pipeline that goes from a diffraction, you know, collection off of the beam to creating a model, we need to be considering more than just the protein information. We need to be considering the solvent information and the ligand information, and get this all together. And we do need metrics such as R-free or others that are considering all of the experimental data and including all of the noise. So we've also sort of expanded and taken this into being able to model a lot of the ligands. This was led by a really talented research assistant in the Fraser lab before I had moved to Vanderbilt. And so we wanted to, you know, ask ways, how can we be able to fit ligands in the same way? And this was building off a previous method that, again, sort of failed in some very specific ways because of sampling. And so I think that this is a really illustrative way that we can see how, if we can improve sampling, that we oftentimes can improve some of this fit. And so what Jessica did is she turned to the RD kit, the experimental torsion knowledge distance geometry conformer generator, which can generate conformations, looking at the longest and shortest distance between every atom. because we were looking at experimental data, we also restricted sort of the sampling in very specific ways. So looking at terminal restraints, sort of blob restraints, or fitting within the experimental or fitting within the binding pocket, used this same mixed integer quadratic programming, and was able to really fit a lot of this information, showing that we can improve RSCC to these, but also recapitulate things in diverse different data sets. So showing it in both macrocycles, which are notoriously flexible and difficult to fit into experimental data. And we also translated this into fragment screening data, so using pandas event maps. Okay, so the last piece of this really is standardization, representation, comparison metrics, as I said, and sort of these uncertainty quantification. So, right, we can sit here and we can hammer out and say we've improved our free, but like, how is that actually translating to other biology questions that we're interested in? Right, so if we go back to this conformational states, they all have different lengths and timescales and contribute to function. How are we actually encoding this information so that we can translate a lot of this rich information that we can get from experimental data to being able to translate it to function? So we have to think about sort of how we are encoding this and how we are sort of translating this information. So if we have sort of again, thinking about it in sort of a sidechain backbone manner, you have multiple different states. So you have sort of 3 different states here. Right now, how oftentimes we can do this is we can either use a multi-conformer model. So we can label these A, B, and then C. You could also duplicate the entire model, so an ensemble model, but that gets incredibly heavy in terms of the amount of replicate information that you're taking into account. And so what we've been proposing is thinking about this sort of hierarchical heterogeneity to describe how states are actually related to each other, so that we can understand both how to explain the entire set of experimental data together, but then it's in a very machine readable and human readable state to understand how these different states are related to each other. This is also really important when it comes to this conformational and compositional heterogeneity. So again, thinking about this open bind data. If you are sort of modeling something with a ligand that is only partially bound, obviously when the ligand is there, the phenylalanine is not going to be in that purple state. And I might be able to understand that if I pull it up in PyMol or I pull it up in Coot, but if we want to sort of translate a lot of this into ML, you don't want to obviously have to be manually understanding a lot of this information. And so the same sort of infrastructure could be used to understand and separate out this conformational and compositional heterogeneity. And with that, I will say I'm not going to present on any of the biology, but I am presenting at 1 p.m. on Wednesday on a lot of this stuff, on the downstream stuff. So come if you're interested. There's also like another fantastic group of people that do a lot of sort of methods development and modeling. And thinking about sort of translating this to biology that are there. So I think a lot of people here would be interested in hearing many of the other speakers. And with that, I will thank the lab, thank funding, and happy to take any questions.
Yeah, we're open to questions.
I had a question about this comparison between QFeed and ensemble modeling. Thanks, good. So I use part in past ensemble modeling, and so I wonder, there's plenty of time that we overfit the data with the noise. So is there any metrics very similar that you try to figure it out for the QFeed? I've never used a QFeed, so just
yeah. So I can say that like the ensemble modeling, I don't know exactly where it overfits. I will say that, like, you know how in ensemble modeling sometimes you can get like structures that will just like part of it will just like go off into like Nowheresville, right? That I think is based off of like a distribution of fit of the model. So for example, if you have the majority of the model that fits really well, and then you have some sort of terminal region that does not, ensemble refinement oftentimes goes into sort of craziness, but that can also happen in other subunits. We do see sort of a relationship between the number of different rotamer states that we fit with QFit and a higher RMSF with ensemble refinement, as we would expect. But where exactly like this overfitting, we don't know yet, but like we're trying to figure that out.
You show the comparison between R3 from Ensemble and then just from the whole set of the sets. It would be good to see it because there have been some cases where QFit had a really bad fit. I mean,
Yeah,
look at
totally.
the set.
No, that's the reason we're doing it, right? Like to try to figure out like, you know, are there things from ensemble refinement that we can sort of leverage and figure out where both methods fail, because they both fail in different areas.
We have time for one. Yes.
Really curious about uncertainty quantification for structural ensembles. Like, how can we distinguish between like variants that are supposed to exist? So, like, how can we distinguish entropy in the ensemble versus like generative model being uncertain and like just generating functional?
I don't have a great answer to that. You know, I kind of view this as like, you know, experimental data gives us ground truth now, right? X-ray is not biological fully ground truth, but I think it's a lot closer to ground truth than some of the generative models that we have right now, because of, for a variety of reasons of sort of, you know, what I was asking you, right? Like we have different data, but eventually, you know, we kind of have to thinking about this in a much more integrative way, with the idea being that if a generative model is able to fit multiple pieces of experimental data, then that's going to be a lot closer to what I think is sort of a biological ground truth, and would give us this information on, is it uncertainty in the model versus sort of uncertainty in the data, and that different experimental pieces would be able to fill some of those gaps and holes in.
Question related to uncertainty. So when we talk about modeling conformations and like the dynamics, we mostly think of like coordinates. But like for example, in crystallography, we not only model the coordinates but also like B factors. How does that translate to when we model multiple conformation and even like ensemble? Especially considering like this might be a bit specific to crystallography where like occupancy and B factor can kind of compensate each other, right? How do you address this?
Yeah, so I mean, thinking about this from like a multi-conformer perspective, if we basically remove like the Bayesian information criteria from QFit or like let it model, it would model 10, 15 structures all at very low occupancy. And then it would translate into also having very low B factor. But is that sort of like useful, right? Then you're getting more to an ensemble based method. And, you know, there is a very, it is like a nuanced way. You know, a lot of the ways that we sort of think about this is trying to make much more parsimonious models that enable people to be able to manipulate some of this and coot. If that, like, but that also is a downside to some of it, right? You are capturing, you're not necessarily allowing sort of a method to be able to capture all of the information that you want. And maybe that's not sort of like the ideal way forward, given sort of how this is tracking. But it's, I don't have like a perfect answer, but it's something that we sort of like think about, and we like make certain algorithmic decisions to think about sort of like where do we actually want the conformational or sort of the heterogeneity, the experimental heterogeneity to sit.
The last question is.
As an experimental scientist, what I've seen is that a lot of these possible measures lesson will disappear, like there's like let's say constructive X-ray scattering, or like the ligands or the metals would be just averaged into the modeling, so we were missing the electrons instead of having them. So I was wondering in this case, like especially when we were like averaging constantly, wouldn't the overfitting or advantages in those cases where they're actually missing the electrons in the
Sorry, I missed one piece here. The overfitting.
wouldn't be advantageous if you're actually missing the electron spaces in the experimental case? So they're already biased.
I mean, I would argue that there has to be other like sort of uncertainty metrics, and whether that is sort of like just you have a very large B factor because there's just like an unknown and uncertainty, that would be a better way to model. But then the question is like, how do you actually like translate that into biological information that there is?
Yeah, it is very
Yeah.
from the experimental case.
Yeah.